Hi, this will be the first of two lectures on protein structure. We've been introduced to proteins as one of the four fundamental biopolymers, and now we'll learn about them in a little bit more detail. Specifically, we'll talk about how proteins are made up of amino acids. It's a linear string like beads on a necklace. We'll talk about the peptide bond, which joins amino acids into the protein. We'll talk about forces which govern the shape of folded proteins, and we'll talk specifically about some misconceptions about hydrogen bonding that helps determine protein structure. Before we get into that, I'd like to point these two pictures out to you. We discussed in a previous lecture about how cells are made up of 70% water, 30% biomolecules. This bottom picture in particular is drawn to scale with the proper concentrations. And when you think of 70% water, you might not think about as crowded an interior as that. There's a lot of molecules. This will affect diffusion, and most of the water in the cell is, even though it's 70%, it's very close, within I think 6 molecules of one of these biomolecules. So there's not a lot of free water inside a cell. Another depiction of 70% water is up here in the protein. These are various pictures of protein crystals. We saw that sodium chloride crystal that sort of makes some sense, spheres, oppositely charged, but even complex shapes like proteins can crystallize. And that's pretty amazing, and I think the pictures are pretty beautiful. I have about 4 or 5 slides of review just to remind you about things that we've learned. The first was that opposite charges attract, like charges repel. We'll use that over and over again. This is in regard to electrostatics, and remember this is primarily an enthalpic term of free energy. The second rule was that oil and water don't mix, and that is another simplified way of stating the hydrophobic effect. And these are due mostly to entropic contributions to free energy. And this is a review of what an amino acid looks like. We have the amino group, the alpha group, and the carboxylic acid. Amino acid. Connected to the alpha carbon is the R group, and we have all 20 different R groups. All amino acids have this backbone and this is where the bonds will form for forming a protein structure. This is a reminder about the 20 amino acids. We're not memorizing structures, but we are, again, remembering functional groups. We recall that there are 5 charged and polar amino acids. There are 5 uncharged but polar amino acids. These are the amino acids that are more likely to be involved with water or aqueous phases. These are hydrophobic amino acids, these 10 here. These side chains are nonpolar, and they will tend to cluster in either, for instance, the hydrophobic core of a protein or in the hydrophobic section of a lipid bilayer. So here's an amino acid. They are joined together to form proteins. A protein is nearly always a linear string of amino acids. And we'll see more about this, but again, this is examples of amino acid 1 right here, side chain 1, methionine. This is the peptide bond right here. This is planar. We won't rotate around this. This is the second side chain, second amino acid, third amino acid, fourth amino acid. When they're formed in linear, most of the time the side chains point in opposite directions, so that you can have 2 sides to a single protein. For instance, this side of the peptide chain will be nonpolar, whereas up here it will be polar. So they can it's a property called amphipathic. We do have rotation around 2 bonds. We have rotation between the amino group and the alpha carbon, and we have rotation between the alpha carbon and the carboxyl group. So this is, as depicted, this is an amino acid. In gray we have peptide bond 1, gray peptide bond 2. Remember that these 4 atoms are planar. You do not have any rotation around this single bond because it has partial double bond character. So these phi and psi angles can take on characteristic values. And you can see there's certain structures that are most common. This is a topographical map showing the most common values. The red are the most common, so that the most likely angles for phi to adopt is around minus 110. Here's 0. Here's minus 180. Around minus 110. And for psi, there's 2 structures that we'll learn about, beta sheet and alpha helix. If it's an alpha helix, psi will take on a value of minus 50. If it's a beta sheet, beta sheet, 150 will be a common value. Any of these values are possible. These are just showing the most frequent found in crystal structures of proteins. This is 4 different depictions of the same protein. This is a backbone-only view. It's sort of simple to see, but again, we get fooled to think that there might be some space there. This is a space-filling model where the atoms have their electronic radii filled in, and as we can see, it's quite dense. It's closely packed. This is basically a backbone view also, except that you can see that we've added some depictions of secondary structures. These arrows are beta sheets. This helix is an alpha helix. That helps our eyes draw attention to certain structural features in the protein. And here, this is in the bottom left-hand corner, we have an all-atom trace. This is just the backbones up here. This is all the atoms being depicted. Now, with regard to forces that help govern the shape of proteins, we've got 3 really important contributors here. The first is a salt bridge, an electrostatic interaction, a positive charge and a negative charge of lysine and I think glutamic acid here. And again, opposite charges attract, so this is going to be a positive enthalpic force. Over here we have hydrogen bonds, and as we remember, that's sort of just a special case of electrostatics. You have a partial negative charge here and a partial positive charge here, and so that's an electrostatic interaction. These hydrogen bonds are linear again because you're going to have repulsion from the partial negative charge here and the partial negative charge on the nitrogen. So again, that is an important contributor to the energy of protein folding. And lastly, we have van der Waals interactions. Now, van der Waals actions occur between any 2 atoms, but they're sort of highlighting here hydrophobic interactions of methyl groups, and we'll see how that contributes to the folding energy of a protein in this next slide. Now, this is a schematic on the left here of a protein with the amino acids alternating between hydrophilic and hydrophobic, and as you can see in the folded structure, all the hydrophobic amino acids have gone to the interior of the protein. This is probably the largest energetic component favoring the folded structure. Notice also that we have not buried any of the polar side chains into the interior. This slide depicts an experiment where we take a folded protein, we add a large amount of urea to the solution. Urea is a molecule that disrupts hydrogen bonds and hydrophobic interactions, and the result of that is it will unfold proteins. That's native structure and denatured. So it unfolds the proteins because you don't have either the hydrophobic forces or the hydrogen bonding to cause the structure. And so then we're going to dialyze away the urea, removing it, and so it's back to its normal buffer. And for many proteins, not all of them, but many, the protein refolds back into its originally native state. One thing that this experiment tells you is that it is the linear sequence of amino acids which primarily determines the folded structure of a protein. There can be some complicating issues with regard to kinetics of folding and the production, but for many proteins, it's simply the amino acid sequence. There are no helper proteins. There's nothing instructing it how to fold. It refolds entirely based on the amino acid sequence. That is because thermodynamically that is what it must do. One last point about this experiment. There are misconceptions in your book and in the literature that says that proteins go to the fold to the global minimum of energy. And that is not entirely clear. We'll discuss this a little bit when we talk about protein folding, but all we know is that it's a very stable formation. It's a local energetic minimum. We don't know if it's a global minimum. A second misconception about protein structure folding is you'll see very frequently it mentioned that hydrogen bonding is responsible for the stability of the folded protein. Let's think about that for a little bit. In a protein, you have hundreds of hydrogen bonds, and each hydrogen bond has an energetic value of between 1 and 2 kilocalories. That would suggest that folded proteins are potentially stable by 200 or 300 kilocalories. In fact, folded proteins are only stable by about 10 to 20 kilocalories if you look at the energetic difference between the unfolded and the folded form. And so you ask how that can be. And the answer is that the hydrogen bonding is not a driving force for folding. It does help determine the final structure because you have to satisfy, the final structure has to satisfy all of these hydrogen bond possibilities. If you don't satisfy all of them, you're going to lose out to the unfolded form. I've tried to draw that schematic here. Here's a folded protein. Every atom which can be hydrogen bonded is hydrogen bonded. So that is one correct solution to satisfying the hydrogen bonded problem. In the unfolded state, though, these are my poor attempts to draw molecules of water. That's not to scale, obviously, but I'm just emphasizing the fact that every single atom that can hydrogen bond is hydrogen bound to a molecule of water. If an amino group or carboxylic acid here were not hydrogen bonded, then you would be favoring the unfolded state. So for this to be equal in energy, everything that's hydrogen bonded here has to be hydrogen bonded here. So the way I think about it is hydrogen bonding is not a driving force for going to the folded conformation, but it is a constraint on the final solutions. That means only those folded shapes in which every hydrogen bond is satisfied or occupied or made will be likely to be in contention with the unfolded form. Again, the prime driver, energetic driver of the folded protein structure is the burying of the hydrophobic core in the center. Now, this misconception about hydrogen bonding in proteins also takes place in DNA. Here we have a C and a G base pair, and they're connected by 3 hydrogen bonds, and you will hear over and over that DNA with high CG content is more stable than DNA that has a lot of AT base pairs. A and T are held together by 2 hydrogen bonds. And they say 3 is stronger than 2, and again, that's incorrect reasoning, because when you have DNA that's single-stranded, all of these hydrogen bonds are going to hydrogen bond to water, so it's not a driving force for the folded conformation. It's a specificity determinant in this case, but not a driving force.